Our next presenter is Eric de Sousa, presenting TCRcloud.
Hello, my name is Eric. I'm going to talk about the tool we've created called TCRcloud. So we created this tool because we wanted to create simple visualizations of TCR repertoires, because we wanted to be able to trace big clones across different time points. And then we decided, since there was their community, to use the ADC API, and we use that to validate rearrangements and repertoires before processing them for creating the images. So the tool right now has 3 functions that we can use. The first one is to create this word cloud based on the CDR trees that are in your rearrangements file. So each color represents the V gene that codifies the CDR tree, and the size of the letters are the relative frequency of the CDR trees in your sample. So for example, here we have the alpha and the beta chain of a tumor, and then the matching tumor infiltrating lymphocytes that were expanded for that tumor. And we are able to see what happened. So we see, for example, in the alpha chain, there was a CDR tree that was not very prevalent, and then it gets expanded in vitro. So with TCRcloud, you don't need to keep the colors based on the V genes. If you want, you can specify which CDR3 sequence you want to focus on. So these are the same CDR3 sequences from before, but now I've matched them with the color that I want to highlight them for easier presentation. Then the other function that TCRcloud does is to create these visualizations based on the various diversity metrics that are commonly used when evaluating repertoires. So most of these metrics are common, like the Simpson index or the Shannon index. The more different ones might be the D50 index. Maybe you haven't heard about it. That was developed by Dr. Jinhan at the Hudson Alpha. And then convergence. Convergence here, it's also a term that's not very well defined in repertoires. Here we define convergent as a CDR3 that has more than one nucleotide sequence coding it. So by default, the program builds the legend based on the TCR, the repertoire ID in the rearrangements file, but you can also customize it. So for example, this image was based on the Mark Davis twin studies, and we're comparing the repertoires of one set of twins. Then the final function of TCRcloud is to download rearrangement files based on repertoire files. So for example, you go to AIRR-C and you find a repertoire that might be interesting to you. So you can download the repertoire file from AIRR-C, and then using TCRcloud, you will get the matching rearrangement file, and then you can create either visualization that you want, the cloud or the radar plot. So our tools is available in PyPy. You can easily get it by saying in the command line using pip. Currently, it's only available in Mac and Linux because one of the dependencies doesn't compile on Windows. But if you want to use Windows subsystem for Linux, it will work fine. Tomorrow I have a poster, and I have applications of these tools in some real cases if you want to discuss it. So thank you for the time, and this is my lab, and I want to thank my supervisors, Marcos Moirer and Margarida Gama Carvalho. Thank you. Questions?
Right. Any questions for Eric?
So I'd like to talk to you about integrating that into AIRR-receptor directly. That looks really cool.
Okay, we can talk about it. Yes.
Hi, can you go back to the slide where you were comparing different different samples for analysis?
Here,
The slide before that.
this one.
I think one more where you had those, exactly, this one. So the boxes over here, do they correspond? So my question is, like, when you're comparing samples side by side, like when you're visually looking at it, you want to compare like either the sequence or the color, right? So if it's abundant in one sample versus another, or like, you know, if you're comparing different patients. So
these are the same, it's the same patient. So it's a tumor, and then the lymphocytes expanded in vitro from the same tumor. So we're just wanting to see how the frequency changes based on how it was in the tumor microenvironment, and then how our in vitro expansion affects the repertoire of that tumor.
okay, that makes sense.
Because our idea is to then give these in vitro expanded T cells back to the patient. So we want to see if we're giving back to the patient something that's very different that was originally in the tumor.
Okay, thank you.
Thanks very much, Eric. That was great.